MCAT 2017 Cram Biochemical and Biological Foundations of Living Systems. Passage 24, Neurofibromin mutations in mice populations. As you view the reading of the passage, you'll notice some highlighted snippets of text. What I want you to do is garner meaning from these specific selections in order to answer the questions that follow. Good luck and happy reading. Paragraph 1. Neurofibromin is a protein encoded by the NF1 gene on chromosome 17. Under normal circumstances, neurofibromin acts as a tumor suppressor in nerve cells. Okay, so here we see the NF1 gene being first transcribed into an mRNA transcript, then translated into this protein, which has over 2,000 protein residues. And the way that it's able to act as a tumor suppressor is that the neurofibromin protein acts as a negative regulator of ROS signaling. Okay, so that's just an aside. Certain mutations in the NF1 gene result in dysfunction of neurofibromin. As a result, individuals with these mutations have increased risk of developing certain benign and malignant tumors. I've had much reservation about actually showing this condition in humans because it's a little graphic and too much for people, but you have to remember, if your goal is the medical field, you can't be grossed out by these things, and you can start to slowly desensitize yourself by looking at the images, okay? It's not meant to scar you for life. It's actually meant to enrich your repertoire of images so that when you see such malformations, you can help the people with the condition. That's what you're going to be doing, okay? All right. Paragraph 2. Neurofibromatosis type 1 is a hereditary disease in humans that is caused by a mutation in the NF1 gene. The disease is inherited in a dominant pattern, okay, and manifests itself in the form of skin hyperpigmentation and numerous neurofibromas, a type of benign cutaneous nerve sheath tumor. In rare cases, a neurofibroma may turn into a malignant tumor called a malignant peripheral nerve sheath tumor, MPNST for short. This process is known as malignant transformation, and this is a histological slide of this condition. Paragraph 3. In order to study neurofibromatosis type 1, a mouse model of the disease has been developed. The phenotypic patterns of the disease in the model mice includes neurofibromas and MPNSTs. Remember, this stands for malignant peripheral nerve sheath tumor. Okay? Paragraph 4. A researcher is studying the rate of phenotypic expression of neurofibromatosis in mice with the NF1 mutation. To complete her study, she creates a large population of mice by initially mating a healthy female mouse with a homozygous diseased male mouse, okay? The initial set of offspring is known as the first filial F1 generation, so F1 for short. The following generations mate at random until the population reaches 10,000 mice. The results of the study are stratified between mice with and without the NF1 mutation. The variables included are the number of mice with neurofibromas and the number of mice with MPNSTs, okay? The results of the study are recorded in Table 1. Table 1. Results of phenotypic expression in mice with and without NF1 mutation. If two mice from the F1 generation of the study were crossed, what percentage of their offspring would have neurofibromatosis type 1? Would it be A, 25%, B, 75%, C, 50%, or D, 100%? And while you're arriving at the correct answer choice, just in case you haven't sorted out the definition of F1 in your mind, F1 is short for first filial. Okay, so that's the first filial generation. And filial just simply denotes the generation or generations that come after a parental generation. Okay, so the F1 generation, sometimes referred to as the F1 hybrid, first filial generation, are the offspring resulting from distinctly different parents that are intentionally crossbred. Okay, so these would be like the first set of mice in the experiment to be crossbred to produce offspring. Okay, so then F2 would be the second filial generation, or the generation resulting from a cross between F1 organisms. All right, so that's just a little bit of background. And now that I've overloaded you, I'll give you an additional moment to think about what answer choice you're going to select. All right, let's get into the solution. Okay, so from the passage, the author tells us specifically that the researcher creates a large population of mice by initially mating a healthy female mouse with a diseased male. Okay, so our healthy female, our diseased male. And this is known as the genotypes of the P generation, where P is short for parental. Okay, the genotypes of the F1 generation can be determined using this Punnett square. All of the offspring, as shown here in the F1 generation, will be heterozygous. So they'll be heterozygous and they will have neurofibromatosis type 1, okay? Because you have to remember that it's a dominant mutation. So now what we can do is use another Punnett square to cross two F1 heterozygous mice, okay? And after we do that, we can basically use this cross of the F1 offspring to create an F2 generation to calculate the percentage of their offspring that have the disease. Okay, so they'll either have to be homozygous dominant or heterozygous in order to qualify as having neurofibromatosis type 1. All right, so if two mice from the F1 generation mate, as shown here, then 75% of their offspring will have neurofibromatosis type 1 because 3 out of 4 have it, or that fraction translates into 0.75, which yields a value of 75%. Okay? All right. Within the final population of mice, what is the heterozygous genotype frequency? Is it A, 16%, B, 48%, C, 12%, or D, 36%? I'll give you a moment to think. And in order to arrive at the correct answer, there's a specific principle that you have to be familiar with. All right, hopefully by now you've arrived at the correct answer, and if not, that's totally fine. Let's take a look on how to go about solving this particular problem. This question requires an in-depth understanding of the Hardy-Weinberg Principle, and the Hardy-Weinberg Principle supports two equations. P plus Q is equivalent to 1, and P squared plus 2PQ plus Q squared is also equivalent to 1, where P equals the frequency of the dominant allele in a population, Q equals the frequency of the recessive allele in the population, P squared is equivalent to the percentage of homozygous dominant alleles. Q squared is equal to the percentage of homozygous recessive alleles, and 2PQ is equivalent to the percentage of heterozygous individuals. Okay? All right, so the proportion of the population with a homozygous recessive genotype is equal to Q squared, as we established previously. And this can be used to determine the frequency of the recessive allele Q. Okay, so table 1 here shows that 36%, or 3,600 out of a total number of 10,000 mice of the population are homozygous recessive. Okay, this is Q squared. So now it's going to be easy to solve for Q. We have 36%, or 3,600 out of 10,000, where 10,000 is the summation of the total number of mice in each population, of those with the NF1 mutation and those without. And you have to remember P is assigned to the NF1 mutation because it's dominant. This is mentioned in the passage, okay? And therefore, mice without the mutation can be considered recessive or the Q frequency. So in order to solve for Q, we have that Q is equivalent to the square root of 0.36, or the square root of 36%, which is going to yield 0.6, okay? And the proportion of the population with the heterozygous genotype, again, is going to be equivalent to 2PQ. So in order to arrive at this, we have to solve for P, and we can take this equation to solve for P. So 1 minus 0.6, or the frequency of recessive alleles in the population, is going to give us the frequency of dominant alleles in the population. Okay, so that's going to be equivalent to 0.4. All right. Therefore, 2PQ is equivalent to 2 times 0.6 times 0.4, or 0.48, which can also be expressed as 48%. So the correct answer choice is going to be B, 48%. All right? Okay. A mouse with an NF1 mutation is chosen at random from the final population. What are the odds that the mouse will be homozygous dominant? 1 to 8, 1 to 12, C, 1 to 4, or D, 1 to 3? I'll give you a moment to decide. Be sure to press pause if you need to. All right. Okay, so this question requires an understanding of the Hardy-Weinberg Principle. All right, and previously I described the two equations supported by the Hardy-Weinberg Principle, which is P plus Q is equivalent to 1, and P squared plus 2PQ plus Q squared equals 1. Let's do a quick rundown again. Here, P represents the frequency of the dominant alleles in the population, and so Q represents the frequency of the recessive alleles in the total population being experimented upon, okay? Here, P squared represents the percentage of homozygous dominant individuals, so it involves it's factoring in basically 2 alleles. And here also, this represents the percentage of homozygous recessive individuals in the population. And here, this represents, 2PQ represents the percentage of heterozygous individuals.
Okay, again, 2 alleles under consideration. All right, so the proportion of the population with homozygous recessive genotypes is going to be Q squared. And we can use this to determine the allele frequency for the recessive allele. Table 1 shows that 3,600 out of 10,000, because if you summate 6,400 plus 3,600, you get 10,000, or 36% of the population is homozygous recessive. And the reason why we're finding out the recessive homozygous proportion of the population rather than the dominant proportion or percentage is because this 6,400 factors in p squared plus 2pq. Okay, so it's not just p squared alone. That's why we have to break down q squared first in order to find out the actual value of p or p squared and eventually 2pq. You'll see why later. Okay, so as previously mentioned, 36% or 3,600 out of 10,000 individuals in the population represent the homozygous recessive individuals. Okay, so Q is equal to the square root of 3,600 over 10,000, which simplifies to 0.36. And the square root of 0.36, which will yield Q, is going to be 0.6. Now, we can solve for P, and P is just going to be this equation rearranged. P equals 1 minus Q, so here we have 1 minus the value of Q, which we just figured out right here, and therefore P is equivalent to 0.4. Okay? All right, so let's continue. The mouse chosen in the question has the NF1 mutation, okay? Therefore, it must either be homozygous dominant, as I previously touched upon gently before, or it has to be a heterozygous genotype, all right? So, therefore, the ratio that we're looking to find is going to be p squared to 2pq, okay? So we want to know out of a mouse with an NF1 mutation chosen at random, it either has to be this genotype or this genotype. We want to know what are the odds that mouse will be heterozygous dominant. So when we ask what the odds of something happening is, we represent that first, and then out of as opposed to the odds of it not happening. Okay, so the odds of it happening is going to be p squared or homozygous dominant, as opposed to being heterozygous dominant. Okay, because this represents the possibility of choosing a mouse with the NF1 mutation, but we don't want to know, what is the percentage out of a total. We want to know what are the chances of this happening with the NF1 mutation as opposed to this, okay? So we're not really concerned with this. That's why we write the odds of the homozygous dominant population first. I hope I didn't confuse you. I know what I'm saying. I just hope you understand what I'm saying. And if I don't, just practice. If you don't, not if I don't. If you don't, just practice this a few times and you'll definitely catch on, I promise. So, because we found P, the P squared value is just going to be P squared 0.16, okay? And because we now know the value of P is 0.4 and the value of Q is 0.6, we can figure out 2pq. So that's going to be 0.48. And this ultimately simplifies to an odds ratio of 1 to 3. Okay, so that's our final answer. The odds of choosing a mouse with an NF1 mutation that is homozygous dominant in their genotype is going to be 1 to 3. So you're 3 times as likely to choose someone who's heterozygous. Okay. All right. Hope I didn't confuse you. If I did, I'm sorry, but this is the Hardy-Weinberg Principle in action. This is how you would apply it. Okay. All right. If a mouse with an unknown genotype is crossed with an unaffected mouse and 50% of their offspring have neurofibromatosis type 1, what is the disease status and genotype of the first mouse? Is it going to be A, disease homozygous dominant, B, healthy homozygous recessive, C, healthy heterozygous, or G, disease heterozygous? I'll give you a moment to think and draw Punnett squares, whatever it is that you need to do to arrive at the correct answer. All right, let's delve into a solution alternative. Hopefully by now you are able to press pause and arrive at the correct answer choice. Okay, so selecting the correct answer choice in this instance is definitely going to require a test cross. Okay, so the genotype of the unaffected mouse that's mentioned in the question stem can be reflected as lowercase n, each n representing one allele, one from the father and the mother, okay? All right, and so each of the possible 3 genotypes, whether it's homozygous dominant, homozygous recessive, or heterozygous, will have distinctly different proportions of disease offspring when crossed with a homozygous recessive mouse, which is basically the unaffected mouse, okay? So let's examine each option. When the homozygous recessive mouse is crossed with a homozygous dominant mouse, basically 100% of the offspring are diseased. So answer choice A can be eliminated. As for answer choice B, when a homozygous recessive mouse is crossed with the unaffected, also homozygous recessive mouse, 100% of the offspring are healthy. Okay, they don't have the dominant mutation. All right, now let's look at the test cross with the heterozygous genotype. So when the unaffected homozygous recessive mouse is crossed with the heterozygous healthy mouse or disease, whichever, well, it has to be one of these because it's either you're heterozygous and healthy or heterozygous and disease. Well, let's take a look and see what happens. So we have here that 50% of the offspring are diseased, and in order to have neurofibromatosis type 1, 50% of the offspring genotype of a mouse crossed with an unaffected mouse must be heterozygous, okay? This is the requirement. And if you are heterozygous, you technically are diseased. You cannot be healthy and heterozygous in this instance because the mutation is a dominant mutation, so it will manifest even in heterozygous genetic quantities, if that even makes sense. But I think you understand what I'm trying to say, okay? Basically, the correct answer choice is answer choice D. Okay, so last question. The results of the study show that despite the presence of a mutated NF1 gene, the rate of phenotype expression in the study population for MPNSTs, which is short for malignant peripheral nerve sheath tumors, is only 5%, okay? So what is the name of this phenomenon? Is it going to be A, variable expressivity? Is it B, incomplete dominance? Is it C, incomplete penetrance? Or is it D, leakage? So this question is really a matter of being familiar with definitions. So I'll give you a moment to think, and if you don't know the answer, that's fine. Just sit tight. We'll review each definition in a second. All right, so hopefully you're able to press pause and arrive at the correct answer choice. If not, let's take a look. Okay, so let's sort out the definition for each answer choice option, okay? Expressivity is defined as the degree to which a gene is expressed. Like in classical biology, in undergrad biology, you might have encountered it in the form of, let's say, a flower and the color for pink or something, pink. Along the monochromatic scale, one might be like a heavy hue of pink or a really light pink. So that's an example of variable expressivity, okay? But we'll revisit expressivity again to finally arrive at the correct answer choice. Now, incomplete dominance is a form of intermediate inheritance in which one allele for a specific trait is not completely expressed over its paired alleles. And this results in like a third phenotype in which is expressed as a physical trait due to the combination of both phenotypes expressed. Okay, so that's so confusing. You can also use the example of a flower for this. Actually, the pink flower thing is better exemplified for incomplete dominance. Let's say the organism in question has an allele for a red flower petal color and white. So incomplete dominance is when both are expressed. So the combination of red and white would yield a pink flower, okay? So they're like co-dominant, or one might be a little bit more dominant than the other, all right? Now leakage, that is the movement of genes from one species to another. So it's definitely not going to be leakage, and we can kind of assume that it's not incomplete dominance as well. That's not the case here, okay? And then what we're left with now is variable expressivity and incomplete penetrance. Well, penetrance is defined as the frequency in which a genotype results in a phenotypic expression. And that seems to be the case here because we're told it only shows up like 5% of the time. Okay, so let's sort out incomplete penetrance and variable expressivity once more because the two sound so similar that they can get confused in some of our minds, okay? So expressivity is the extent to which you express genes, all right? Let's say a mouse has the gene for brown fur. It might come out as dark brown, tan, somewhere between tan and brown. That's variable expressivity. While penetrance determines whether you express the gene or not. Okay, so the mouse might not be like colored white, although it has the allele for brown fur. So expressivity, therefore, determines how well you express those genes, okay? Let's sort this out one more time. Expressivity describes the phenomenon of differing clinical features or phenotypes among individuals carrying the same exact gene allele or genotype, all right? Wow, and expressivity definitely differs from penetrance because penetrance describes the probability or the frequency with which that genetic variation or allele will yield a phenotype at all. So I've said it a lot and I've repeated myself a few times. Therefore, the correct answer choice is going to be incomplete penetrance, because the rate of phenotype expression is basically less than 105%. Okay? If you have a specific allele, you would expect that organism to express that phenotype that corresponds to that genotype or allele. But incomplete penetrance occurs when the rate of phenotype expression is anything less than 100 for a specific genotype. All right? Okay.